Okay, right. Okay, good afternoon. Can you hear me? Yes, can you hear me? Okay, right. So we'll start in another maybe 3 minutes. Okay, because people are still joining. People are waking up from their rainy day naps, and okay, so we'll join in another few more minutes. Okay, right. Shall we start? Okay. So it's paper number 5, right? So, what about the paper 4? Are we, I mean, have we completely discussed it, right? Paper 4 is completely done, right? What's the situation there? Yeah, paper 4 we completed, right? Any issues you had? Yeah. So yeah, I think it's completed, no? Right? Okay. Cartilage. Other than yeah, this was done last week. We completely finished it, right? Paper 4, is it completely done? Yeah. So okay. So today we'll take the fresh start into the new paper. That is paper 5. So as usual, we have MCQs and we have SCQs. Okay. So any you're not facing any weather related problems, right? Anyone who's affected any issues? No, right? Okay. Any power cuts or anything? Not yet. Neither. We don't know what can happen. So let's see. As usual, I mean, so the theory classes are going ahead. So in the theory, we will finish nutrition this week. Uh, Nutrition together with so digestive system. We are in the final part of digestive system, and we'll finish that, and we'll completely conclude. So I'm going to start including questions from nutrition in the paper class, in the papers of the paper class. Okay. So as you know, papers are going to be constantly sort of expanded into, I mean, with the latest theory part that we are doing in the theory class. Okay. So that's the purpose of, it's sort of our specialty. We are not going to stick to these simple, I mean, not simple, right? Earlier lessons again and again and again, because you know it's at the end it is the animal form and the sort of plant form and animal form, then upcoming microbiology, those kind of lessons are going to build, I'm going to sort of contribute to the bulk of the marks, right? So it is important that we start adding animal form questions as early as possible to the papers that we are doing. So that's going to sort of sort of to I mean to enhance and to sort of confirm and reinforce your animal form knowledge early on. Okay, right. So anyway, we'll start. First question: growth and development are characteristic features of living organisms. Which of the following is correct regarding growth? Hari. Quantitative increment that occurs reversibly. What do you think? Growth is always irreversible. Right? It is always associated with development, of course, right? So it is not something reversible, therefore it's false. All living organisms show growth throughout their lifetime. What do you think? Throughout their lifetime? That is a bit doubtful. Okay, throughout their lifetime is a bit doubtful. So let's keep that. Microbes have a high rate of growth. That is correct. Okay, microbes have a high rate of growth. So I'm going to need answers in the chat. Nothing in this rainy weather. Okay. Okay, growth is a characteristic shown only by organisms. Growth is a characteristic shown only by organisms. What do you think? No, no. Right? Things can grow. Right? What are the things that are non-living? Right? What are the things that are non-living that can grow? Right? If you go to a salt and there are salt crystals that can grow. Right? And if you go to these caves, you might have seen there are sort of there are hanging sort of salt sort of cones again, stalactites and stalagmites kilagina, right? I don't think I'm pronouncing it as stalactites and stalactites. Again, may hanging, there's another name. There's this sort of other cones on the ground. Right? But caves are there, right? On the cave roof, there are some sort of mineralized structures that are hanging down, and there are things that are growing from the ground as well. These are minerals, right? Mineralized. So sort of mineralized salt related structures, they can get bigger in size, but they are non-living. So there is a growth, but growth and development are both associated only living organisms. Okay. Um, Cell division takes place as a step of growth in all living organisms. No, I mean proper cell division kineka is not observed in all organisms, right? Some organisms have some sort of how they multiply is by divisions such as binary fission, right? You can remember they have sort of your prokaryotic organisms, they multiply by binary fission, right? Okay, then there are other things such as budding and fragmentation, right? Those kind of things. Yeah, growth is a characteristic shown only by organisms. The organisms it is, yes, it is living organisms, right? Nothing. There are dead organisms that we don't call them. I mean, they are anyway, they are decomposing, right? Organisms is living organisms. So the best answer is answer number 3. Microbes have a high rate of growth. Okay, that is because this is something you will talk about when you are doing microbiology. Um, Microbes have a high ratio of surface area to volume. That means their body surface area is very high compared to their volume. Okay, so that means if you take a typical microorganism, they have a lot of surface area for the entry of nutrition and other things, and also that same surface area is going to help these microorganisms to sort of they have this cilia, right? And this they can have, I mean, their surface area is very high. Okay, so therefore they have a larger surface area to take in nutrients and gases and also to excrete things. Okay, therefore they have a high rate of growth because their metabolism is high, because their metabolism is high. Okay, so best answer is answer number 3. Haridha, right? Which of the following is correct regarding protein? Protein. Sulfur is always included as a component element. Not always, right? I mean, hold on. They ask you about protein, right? Yes, I mean, always sulfur is a component. But if you modify this, you can correct regarding amino acids. Is it correct? Do you always have sulfur in amino acids? Do you always have sulfur in amino acids? No. Right? If you take your simplest amino acid, that is your glycine, these are basic things. Right? COH and your NH2. Right? If you take your glycine, it doesn't have any sulfur. Neither. So all amino acids doesn't have sulfur, but all proteins have sulfur because we talked about the elemental composition of proteins: carbon, hydrogen, oxygen, nitrogen, and sulfur. Neither. Those are the elemental that are sort of elements that are involved in uh, protein formation, right? Okay. Then next statement. All bonds break down at denaturation. No, no, that is not correct. What do you think? All bonds can break down up to the level of peptide bonds, right? So you are sort of definitely hydrogen bonds. So your secondary structure is composed of hydrogen bonds, right? So your hydrogen bonds definitely break down. Tertiary structure has one other kianaban. Send me in the chat what are the bonds in the tertiary structure. These are basic things. And 2028 set එක to ගන්නනේ මේවා. Yeah, what do you have? Disulfide bonds, yes. Ionic bonds, van der Waals interactions. Anything else? I'm forgetting. Disulfide, van der Waals, hydrogen, yeah, say we said. What else? Those are the things, no? Ionic bonds, right? So all those bonds can be broken down. So tertiary structure, secondary structure, all those things can be destroyed, but the primary structure remains. So the peptide bonds are not broken down. Usually in denaturation, peptide bonds are not broken down. So all bonds break, it is false. Got it? Collagen protein is a tertiary structure. Collagen has how many protein subunits? I think if you can remember, in early days, Tutikale, I gave you a diagram of collagen as well. Collagen has three protein subunits. Okay, collagen has three protein subunits. I mean, so it looks like three strands. Okay, so protein collagen has three. If you have more than one subunit, I mean, that is more than one polypeptide chain that are forming a protein, that means it's a make correct karana. You can write a review, right? It's a quaternary structure, right? You can correct this, right? Collagen is a quaternary structure, which protein with quaternary structure. It provides strength, yes. Then I'm keep on telling in your A level questions, an MCQ statement has, I mean, it contains several ideas, right? Several ideas can be there in a A level question MCQ stem. That is because, I mean, you can see here we are asking if it's what is its structure and what is its function both. So one can be correct, but one can be wrong. So the correct one is mostly put there to confuse you. Like confused they are putting the incorrect statement in there as, sorry, the correct one. So මේක දැක්කපු ගමන් මේකත් හරි කියලා it comes into your subconsciousness, right? Rapid dissociation of regulatory protein is a must for their functions. Yes, a regulatory protein such as, for example, what do you mean by a regulatory protein? What are the regulatory proteins? Proteins having regulatory functions. There are the proteins, there's a function chart. There are protective proteins, transport proteins, structural proteins, yeah, catalytic proteins. So regulatory proteins are mostly what? Your hormones, your peptide hormones, yeah. So the hormones are like a signal, right? If you want to, yeah, that is those are examples. Yes, hormones, right? A regulatory protein shouldn't be something that is difficult to remove. I mean, sometimes it can be difficult to remove, right? A regulatory protein should be able to bind fast and also release fast. That means, I mean, it can give the signal and it can remove the signal fast, right? Therefore, a regulatory protein, you can write here example hormones. Yes, that is a very good correct statement. Okay, hormones must be able to dissociate rapidly. Fibrinogen is a contractile protein involved in blood clotting. Fibrinogen is a contractile protein involved in blood clotting. What do you think? Then there are several ideas, right? You don't know exactly fibrinogen yet. Okay, fibrinogen is involved in blood clotting, that is correct, right? But it's not a contractile protein, it's a clotting factor, right? But you can answer this one question because you know what are the contractile proteins. What are the contractile proteins that you have? Yeah, what are the contractile proteins? Those are myosin and actin. Those are myosin and actin. So fibrinogen is a blood protein involved in blood clotting, yes, but it's not a catalytic protein. Yeah, got it? Clear? Shall we move on to MCQ number 3? Which of the following combination is true regarding vesicles in eukaryotic cytoplasms and their functions, right? So eukaryotic, there are some important terms, right? So it has to be eukaryotic cytoplasm. Okay, so think about eukaryotic cells. Let's see. Vesicle and function. Glyoxysomes possess oxidative enzymes. What do you think? Glyoxysomes possess oxidative enzymes? What do you think? Yeah, a glyoxysome is a type of peroxisome. A glyoxysome, you can write there, a glyoxysome is a specialized peroxisome. A glyoxysome is a specialized peroxisome. Therefore, it does contain, because peroxisomes contain oxidative enzymes. So, that is correct. Peroxisomes involved in detoxification? Yes, detoxification of peroxides. No, peroxides are toxic. No, peroxides are toxic. Therefore, they have to be removed. Otherwise, they can oxidize other things. They can oxidize other proteins, damaging, causing damage to structure. So that is correct. Okay. Then lysosomes transport of residual material out of the cell by exocytosis. Hi. The make a E1 2028 at a kilo. Can look right? What are the four functions of lysosomes in the cell? What are the four functions of lysosome in the cell? What are the four functions of lysosomes in a cell? Yeah. What are the four functions? You have one of these is given here, right? So residual material, how do you take the residual material inside? How do you take residual material inside? That is by cell eating, no? So digest food particles taken in by phagocytosis. නේද? Digest food particles taken by අපි මේ කතා කරන්නේ මොකක් ගැනද? We are talking about functions of lysosomes. Right? Digest food particles taken in by phagocytosis. Right? Then this is the second function, that is, so after you have eaten, you have to remove the waste. After you have lunch, you remove the remaining malu katu and all the other things, no? Right? So it's like that. So after the cell has digested something, it can remove it. It should remove that out of the cell. Cell like clean karan. So that is the second one. Anyone remember what are the other two? Anyone remember what are the other two functions? Yes, digest worn out organelles. Okay, worn out organelles. That is called autophagy, but you don't need that, right? So when the organelles are too old, for example, if a mitochondria is not working very properly because its enzymes are damaged, it's very old, right? So it's better to recycle the things that are in the mitochondria and make a new mitochondria. So you have to break down that particular organelle so that you can make a new one. Nikan vehicle like engine එක පරණාම you put a new engine. It's like that. Okay. Finally, when a cell is too damaged, right? There are complex mechanisms. When a cell is too damaged, for example, maybe its DNA is damaged or it has been damaged by other things. The cell decides, okay, it is better to commit suicide other than living because there's a risk of it becoming an abnormal cell, cancer cell එකක් වෙන්න පුළුවන්. So that is called autolysis. තමන්වම lyse කරගන්නවා. That is autolysis causing cell death. Yeah, that is very good. So best answer is answer number 4 නේද? ඔක්කොම හරිනේ. Answer number 4 is good. Okay, all right. Right. What's the question? And I take it correct even though peroxide isn't mentioned in B. What do you need to? You don't need to mention peroxides because peroxisomes contain. Peroxides are the things that are broken down by peroxisomes. That is the definition, right? Because they contain oxidative enzymes. They remove peroxides. One of the main functions of peroxisomes. Okay. Right. What about this one? A diploid cell with 8 chromosomes. Okay. Whenever you are reading this sort of questions with sort of certain figures, then physics, chemistry, you do this, right? You underline the important figures, right? You underline, no, highlight, or put circle, or whatever. Okay. Undergoes meiosis. Okay. How many DNA molecules exist in this cell when it enters into the initiative phase of division, in which chromosome numbers are halved? Okay. What do you think? So, how many DNA molecules exist when it enters into the initiative phase of division? Which chromosomes numbers are halved. So they are asking how many DNA do you have when it enters, I mean, before its chromosome number become half, right? So you have a mother cell, right? So at the end of meiosis 1, what's going to happen? Then this divides into 2 more. So you are going to have, at the end of meiosis 2, you are going to have 4 cells, no? Right? So a diploid cell, so imagine this is the cell before it enters into the cell cycle. So it has 8 chromosomes. So 8, 2n is equal to 8. 2n is equal to 8, no? Right? Diploid cell has 8 chromosomes. Then you need to know, so at the end you are going to have n, no? Haploid one at the end of the meiosis cycle. So n is equal to 4. N is equal to 4 haploid. N is equal to 4. Right? Nothing serious, right? At the end of meiosis 1, you know, meiosis 1 at the end you have become haploid, no? At the end of meiosis 1 you have become haploid. Why is that? Because you separated the sister the sorry you separated the homologs. So as soon as you separated this homologs you have become haploid. Okay, therefore what is the cell division in which chromosome number is halved? What is the cell division in which the chromosome number is halved? That is meiosis 1. Right? Meiosis 1, you already have half the chromosome number. But what's the tricky question? What's the tricky part? This cell, when it enters into the cell cycle, how many chromosomes does it have? Because it undergoes S phase, no? Right? It undergoes S phase. So when in S phase, what's going to happen? Yeah, the DNA amount is going to be doubled. Therefore, all the chromosomes are going to be doubled, right? All the chromosomes are going to be doubled. So this person, this cell is going to have 16 chromosomes when it enters into the cell cycle. Yeah. So when it is undergoes the mitotic phase, right? So when it enters into the chromosome number halving phase, right? So it's going to have 8 each, right? It's going to have 8 each. Then once you, I mean, chromosome, it's going to have 8 DNA molecules, but they are double DNA molecules, no? So because they are in the still in the form of sister chromatids. So once they separate into the two individuals, I mean, when the sisters are separated, you are going to have 4 each, right? You are going to have 4 each. But if you consider the number of DNA molecules, right? The number of DNA molecules they are going to have is 8. But that DNA is actually, I mean, atrama hatarai DNA, but they are still doubled. So because of that doubling, they are going to have 8. Got it? Clear? Okay. Okay, sorry. Right. Let's move on to the next question. Question number 6. Which of the following phenomenal combinations is correct? Okay, right. So on Monday, we have this sort of a community medicine new appointment that we are starting. Therefore, I am the coordinator there. I keep getting calls for that, but anyway, it doesn't matter to you. Okay, which of the following phenomenal combinations is correct? Photorespiration occurs in all organisms. No, even not in all plants, right? Not even in all plants. In C4 plants, you don't have photorespiration happening, right? So definitely that's out. Okay. Then what else? Photosynthesis is an anabolic reaction which occurs only in chloroplast. Then again, I told you. Uh, Fifth question. I'm sorry. Right. Hari make kallembu sixth ka patangatte. Right. Photosynthesis is an anabolic reaction. Anabolism means anala hadano, no? Right. You are making synthesizing something. Yes. So it is an anabolic reaction. You use simple things. For example, you take carbon dioxide and you convert it into organic food in the glucose, right? So that is the most basic form of photosynthesis. No? Yes. So it's an anabolic reaction. What about this part? Is it occurring only in chloroplast? What do you think? Is it occurring only in chloroplast? No. There are photosynthetic prokaryotes. Ne? There are photosynthetic prokaryotes that do not have any, they don't have any chloroplast, right? Chloroplast is something only found in eukaryotic cells. So they have membranes that do not show any stacking. Matada dangara chart, right? Cellular basis, that prokaryotic eukaryotic comparison chart, right? Prokaryotes have membranes that do not show any stacking modified, I mean, that perform photosynthesis. So chloroplast is not always needed. Okay, it occurs even in prokaryotic cells. Phototropism always promotes photosynthesis. If you can correct this, the message if you can correct this statement, how would you correct this? If you can correct this, how would you correct? How would you correct? There are two types of phototropism. There are two types of phototropism. Imagine this is the sun, right? Imagine this is the sun. Okay, there are two types of phototropism. There can be a shoot that is growing towards the sun, or there can be a shoot that is growing away from the sun. Yeah. So this is called positive phototropism. This is called negative phototropism. No? So only positive phototropism promotes photosynthesis, right? So sometimes there's a variation, very rarely, right? So you can modify this. You can say positive phototropism promotes photosynthesis, yes, but not all phototropisms. Photoprotection, dissipation of excessive light energy only. What do you think? What are the functions of, why do you do photo, what is the main purpose of doing photo protection, and who does photo protection? Basic things. Photo protection by what pigments? Photo protection is done by what pigments? What kind of pigments? They are done by Ken. Who's involved in photoprotection? Israel carotenoids. The carotenoids. Some of the carotenoids are involved in photoprotection. Okay. So absorption and dissipation of excessive light energy. Otherwise, this light energy may interact with oxygen and form oxidative, reactive oxidative compounds, which can be dangerous, right? So that's okay, right? That is basically photoprotection. What do you think about this photoreceptors? Phytochromes. So you know there are two types of photoreceptors. No, there are cryptochromes and phytochromes. Okay, what is done by your phytochromes? Provide the plant with information about the quality of life. Sorry, quality of light. What do you think? Phytochromes. Phytochromes, they do. right? Under plant spacing, phytochromes provide the plant with information about the quality of light. Yes. So therefore, the plant can detect if it's receiving direct sunlight or if it's only receiving filtered sunlight. That's how the plant understands if it's a emergent tree that is above the forest canopy or if it's someone that is below the forest canopy. That's how the plant understands where am I in the location. Social classes okay, then middle class, upper class, then top sort of the higher upper class, I mean, right? So in our society, it's like that again. If you take a tropical rainforest, there are stratifications, plant layers. So that's how the plant understands, a tree understands where it's in this stratification layers. So yes, phytochromes do provide information about the quality of light. And photoprotection is the absorption and dissipation. Wait. Why is it false? The Meganang, it's a bit of because I took this from a school paper, so it's a bit excessive. Not very, not a very logical question. So photoprotection is not only dissipation, not only just dissipation, but is missing here. Dissipation plus absorption is also there, right? So photoprotection is dissipation and absorption of excessive light energy. Okay, so therefore we can't take 3 as a correct answer, sorry, 4 as a correct answer, 5 is the best answer because they provide information about the quality of light. What do you mean? Cryptochromes about the quality of light? No, right? Cryptochromes don't do they provide anything about the quality of light? No, right? So see, germination is also phytochromes. Cryptochromes doesn't have anything regarding sort of the quality of light. Maybe flowering. Flowering can be controlled by no wait. Again, there is also phytochromes. Cryptochromes has very limited functions. Okay. Yeah, this is correct. Answer number 5. Right. අපි යමු fifth එකට මේක නිකන් දාලා ගියපු MCQ එක, right? Okay. Got it? At enzyme reactions, so right, they're asking the correct statement probably, right? Okay, right. And okay, so what is given? A enzymes are not being used up, of course. They are not reactants. Your enzymes are not reactants. They are simply what? They are simply enzymes are what? You learn about this in chemistry as well. They are simply catalysts. Catalysts are not used up. Yeah, that is correct. Substrate is the reactant. Yes, substrate is a reactant. Okay, that's what gets converted to the product, right? So substrate gets converted to the product. So it is a reactant. Many reactions can be catalyzed by one type of enzyme, not exactly, right? Most enzyme catalyzed reactions are specific to that enzyme, right? So most enzyme catalyzed reactions are enzyme specific. Otherwise, it defeats the purpose of having different enzymes. And for example, if one enzyme catalyzes several reactions, that means there's no proper regulation. That is not very good. So if you want proper regulation, that means you should have separate enzymes or separate reactions. So usually, one enzyme controls a specific reaction, controls and catalyzes. Okay, enzymes do not alter the nature or the properties of the end product. Yes, then you go. Whether you have the enzyme or not, the end product is the same. The enzyme is simply it pushes the reaction. I mean, please do the reaction. It's there to support the reaction. It is not there to change whatever is happening at the reaction, right? Okay, right. So I got caught in the rain, so the results are coming out. Okay, need high concentrations of enzymes for catalyzing a reaction? No, it's a catalyst. A catalyst, even a very small amount, is highly potent, right? It's very effective. A very small amount of a catalyst is very effective. So A, B, D, no? Answer number 2. ABD. Okay, simple question. Okay, right, moving on. Question number 7, right? The questions are a bit easy, today. So for the in the theory class, I plan to have a term test. Okay. So when are your term tests? Right. There was a request to have another term test because we couldn't have one at the end of evolution or at the end of plant form. So I am going to have a proper exam with your papers marked, and during the theory class time, you can write the papers. Right. So exam day is going to happen. I'll let you know. Probably next month. So this today is 15th, no? So we have only 2 more weeks, and maybe the final week of this month. Let's see, right? So it is not going to include any animal form, only from plant form, evolution, and cellular basis. We'll have a paper, and that paper will be marked and given. Online people can join during join at the theory class time and to the live Zoom, and the paper will be sent to you at that moment, and you can answer the paper. Okay, so we'll start. I mean, that will happen. I'll let you know the exact date, maybe 1 week before that we are going to have an exam. Okay, when are your term tests? Terms is kawatina. Yeah, when are your term tests? That is a grade 12 final term test, right? Oh, hold on. What's the term test that is coming up? Grade 12 final one? No, yeah, no. Grade 12 third term test, right? Send me a response. Yeah. June 2nd, very close. July. Okay. Second term. Okay. Right. Very
Exam
close.
July ini.
July
Minggu lagi
last
term
week.
test
Okay,
Julai.
then let's have one right now. I mean, at the end of this month or early next month, we can have an exam, sort of preparing you. Okay, right. So you can write it at the at ISM, and it will be, you can join a Zoom and write live in the live class as well. Hari, that will be updated in the group, okay? And yeah, another announcement: people who are watching, going to watch the recording or here live, do not mute the groups. I have sort of informed this for my 2028 people because they are constantly bothered. They are being added to different teachers groups all the time. Okay, so they are they have a habit of muting all the groups. Even in the groups, they are going to classes, right? So my advice is, do not mute. I mean, you can mute or whatever, but do not miss any updates from our groups, right? So ISM people, there's a ISM 2027 group, and there's a main group for the online people, right? That's the two groups we are having now. So I put all the updates, all the papers, anything additional, and all the class recordings and whatever into those groups. So do not, I mean, all the updates are there. Okay, so keep the groups. I mean, because no one else can message them, so there's very little spam in those groups. Only me and my hotline can message. So yeah, keep those groups. I mean, be aware of those groups, whatever that is coming. Hari. Select the incorrect statement regarding glycolysis. Hari, occurs in all cells, right? Glycolysis occurs in all cells. What do you think? Does glycolysis? So they are asking incorrect. Hari, they are asking incorrect. Yes, glycolysis is an energy producing mechanism that is common to all cells, whether aerobic or anaerobic, whatever. You have to do some sort of energy making, no? So that is done by glycolysis. So that is incorrect. No, incorrect. No, that is correct. Produces 4 ATP molecules by substrate phosphorylation. Yes, you break down complex thing into simple molecules. At the process, you are releasing energy that is going to produce ATP. Very correct. Oxidation of 2 NAD plus into 2 NADH molecules. The mega, again, you are testing two theory points in one statement. Yes, you are converting NAD plus into NADH, but is it a oxidation? Is it an oxidation? No, you are reducing NAD plus. Yeah, හොඳට කියවන්න ඕන කියන්නේ ඒකයි, right? NAD plus plus electrons plus H2.
හරි වගේ තේනවා
Having a series
මුල
of enzyme
වැරද්දක්
reactions. Uh,
කියලා දෙනවා හොඳට මේ
Enzyme reactions, yes, there are many enzymes involved. Okay, anyone else talking? Okay, unmute. Okay, right. Now okay, right? So, series of enzyme reactions. So there is glycolysis. Okay, initiating always only with the 6 carbon molecule. No, glycolysis can have other substrates, right? Glycolysis can have other substrates. Fatty acids can enter into the glycolysis, right? And protein breakdown products can enter into glycolysis. Or not always it starts with the meaning glucose. No, so not always glucose. So actually C and E are the incorrect. D, having a series of enzyme reactions. So that question is a bit doubtful. Okay. So if you have taken C, D, E, that is the answer, but D, that is somewhat correct, right? Glycolysis is a series of enzyme catalyzed reactions. Okay. So don't worry about this one. Okay. This is actually kind of correct, but the best answer is C, D, E. Okay. Okay. Right. Question number 8. Let's move on to question number 8. Incorrect regarding binomial nomenclature. Name of the organism possesses two words. Yes. So they are asking incorrect one. It's binomial nomenclature. Ne? Bi means two, nomial means two, I mean the name. So two name, binomial, it has two parts, two names. First word is a generic name which species include in. What do you think? It is, so in the species that you are naming, first name is, so the first word is the genus of the species. Particular animal ke genus is indicated, I mean written by the first part. Yeah, that is correct. Species with a close relationship possess the same specific epithet? No. The specific epithet is specific. A certain species has only, I mean, the specific epithet is specific to a specific organism. Okay, it is specific, right? So if you have two closely related organisms, they are going to share what? They are going to share the generic name, but two will never have the same specific epithet. So that is loku varadda, right? Big wrong. So it's the correct answer. It is Latinized, right? Latinized. Then Ceylon කියලා ලියනකොට ශ්‍රී ලංක Dipterocarpus zelanicus කියලා අපි ලියන්නේ. Dipterocarpus Sri Lankan මෙහෙම ලියන්නේ. We have to italicize, right? We have to convert whatever we are trying to say into Italy, sort of it, I mean sorry, Latinized. I'm sorry. You have to sort of Latinize the idea that we are trying to give. So that's what that's one of the rules of binomial nomenclature. Should be underlined when handwritten? Yes, of course. Italicized when typed, underlined when handwritten. These are simple, right? Okay. So next question. Right. Which of the following statements regarding seed germination is incorrect? මේ paper එකේ ඔක්කොම incorrect ඒවා අහන්නේ, right? So red and far red increase the percentage of seed germination. What do you think? Red and far red increase the percentage of seed germination. What do you think? Mema, right? For seed germination, what do you need? So, even though these are both red shades, what is the light that is going to actually stimulate seed germination? That is only red, no? Far red means infrared. So, infrared is going to actually inhibit seed germination, right? It is going to actually inhibit seed germination. So, that is not correct. Take a part, right? We have the incorrect statement, answer number 1. Got it? Then, but anyway, we'll go through the other statements. Elongation of hypocotyl slows during seed germination when a seedling breaks ground by blue light photoreceptors. Yes, light induced slowing of hypocotyl elongation is under the cryptochrome control. Cryptochrome, it is controlled by blue light. That is one of the sort of textbook theory parts, right? Why you need to slow down the hypocotyl elongation? I have talked about this again and again and again. um, Whenever a seed, so for most of the time the seed is probably underground, right? It has fallen down and it sort of goes under the sea, under the soil with time. So when it starts to germinate, usually slowly a small seedling, sort of small leaf and sort of the bud sort of grows out of the soil, and so it is exposed to the sunlight. So until that small, that seedling breaks ground, the seed focuses on getting tall. Because why? It needs to reach the sunlight as soon as possible. If it didn't reach sunlight as soon as possible, if it cannot start doing photosynthesis. If it doesn't start doing photosynthesis immediately, there's a risk that it might run out of stored food. If it runs out of stored food, then that is the end of the story. Because if you take, like, but especially small seeds, they have only a very small amount of stored food, right? So they have to reach sunlight as soon as possible. But as so once they have broken the ground, so they are exposed to sunlight, what is now their focus? They shouldn't grow any taller right now. It's not the immediate priority, right? So the immediate priority is to produce more and more leaves, to increase the surface area, to spread sort of laterally, no, to become more wide. That is going to enhance photosynthesis. So that's why as soon as it is exposed to sunlight, the blue light in that sunlight is going to inhibit its growing further taller temporarily. Okay. Gibberellin simulates seed germination as well as seed development. Yes, this is one of the functions of gibberellin that we have included in the list of functions. Right? Abscisic acid inhibits early germination of seed. What do you think? Yes, usually abscisic acid. If you have abscisic acid in your system, if you have a abscisic acid in the seed, that means the plant or the seed is facing a sort of a stressful condition. Abscisic acid, I told you, abscisic acid and these ethylene and other thing, the ethylene and abscisic acid, they are stressful. They are stress. I mean, they are released in response to stress. They are sort of negative plant growth substances. So they promote seed dormancy. They sort of he make the seed stay sleepy or stay inactive for a longer time. So abscisic acid inhibits, yes. Gravitropism starts as soon as a seed germination takes place. Yes, the seed should detect which way is the ground, which way is the soil, which way is the upwards. Right? For example, imagine you have a seedling that is on the ground. The mehema, this is the soil, this is the soil. Okay. So you have a seed. Okay. So you have a seed, chuti abhidamu, sort of a small rice seed that is here. Right? So it is going to project us very small. It is going to body seedling sort of a small growth that growth should immediately detect which way is up and which way is down. Why? If it doesn't detect which way is up, it's the sort of the plumule that is going to become the shoot is not going to grow upwards towards the sunlight. Then if it doesn't detect which way is down, it's not going to extend the radical properly to become the root system. Got it? So it has to detect gravitropism, I mean the gravity, as soon as possible. Otherwise, it cannot extend properly. Shoots upwards, roots downwards. Okay? So it has to start as soon as possible. It has to start as soon as possible. Otherwise, the seed cannot grow properly. Um, Far-red and infrared is the same. So in bio, we call infrared as far-red, right? So in the visible spectrum, no? Right? In the visible spectrum, after red, you have what? I mean, sort of before red, ne? Before red, towards your sort of radio waves. What do you have? Your infrared, because it has a longer wavelength than red, no? So in bio, we call it far-red, okay? Infrared or far-red. Clear? Okay. Question number 10. Again, I think the correct statement regarding the tissue which involved in homeostasis. Okay, homeostasis. What do you know about homeostasis? That is how we stay alive. Actually, right? There are so many things in our body which is like a complex machine. Right? If you take a car, what are the things that you have to maintain in a car? What are the things, conditions that you have to maintain at a normal level in a car? If you think about the engine, then පොඩ්ඩක් engine ගැන දැනගන්න, right? Any sort of a machine, what do you have to maintain? You have to keep the machine cool, no? You shouldn't allow a machine or an engine, whatever, even a laptop or whatever, you shouldn't allow it to get too hot. What else? If it's an engine, you have to provide it with petrol, you have to provide it with engine oil, right? You have to provide it with coolant, right? All those things should happen. That is because that engine is running at optimum condition when the, I mean, sort of optimum efficiency when the conditions are optimal, ne? It should run, it should have a very, I mean, so each engine has a optimum temperature for its performance, maximum performance, right? It should have oil pressure and everything. Similarly, your body, which is like a complex machine, has some certain set of parameters. If those parameters are not properly maintained, the body is not going to work very well. So our body has several dedicated mechanisms to maintain those parameters, because the parameters can have certain conditions. What are the examples? Blood glucose level, body temperature. What else? Yeah, energy availability, that is determined by blood glucose level. Okay. So, I mean, those are the main homeostatic mechanisms. Your heat gain and heat loss, that is coming under maintenance of body temperature. Okay. So, those mechanisms maintain our internal environment at a very favorable level for the body processes. Body pH, right? In our blood, there's a pH level that has to be maintained. Blood osmolality, right? That means the concentration of ions and water in our blood, in our body fluids, tissues. All those things should be maintained in a proper range, otherwise our body will not work as a proper machine. So that is what we call as homeostasis. Okay, homeostasis. So, okay, tissue which involved in homeostasis, right? Let's see. So what is the homeostasis, main tissue involved in our homeostasis? What do you think? There are two systems that are involved in our homeostasis, that is the hormonal or endocrine and the nervous. Okay, so both are involved in homeostasis. Hormonal is mainly involved in sort of a slower type of homeostasis. Nervous system has a very fast response. Functional and structural unit is the neuron. What do you think? So we are going to focus on the neuron neurons. So we are going to focus on this system of the homeostasis. Okay, if you take the nervous tissue, what is the functional unit of the nervous tissue? A nervous system is not just one neuronal cell, right? Then, for example, if you take if you ask what is the structural and functional unit of life, basic, it is the cell, no? Because if you take you and if you take one of your cells, you can say this cell is the smallest thing in your body that you can call as living. So therefore it is a functional unit of life, as well as it is what building up your body, so it's also the structural unit. But if you take a nervous system always has an input, processor, output. Or body ka light ikiran kannukku killa denavane. Like even in the body, you should have an input that is detected by a type of nerve that is called the sensory nerve, right? Right? Then that sensory input will be processed by another nerve that is part of the central nervous system, that is called the interneuron, no? Sensory neuron to interneuron. Then there will be a response that is generated by this processor that will be taken to a sort of a effector organ. It can do something. So that effector organ is going to be supplied by a motor neuron. That effector organ can be a muscle, another organ, a gland, whatever. Okay, this is called Anyone know what this called? There's an input and a processor and an output. What do you call this system? What do you call this system? Anybody know? Anybody know? What is this? It's the simplest working model of the nervous system. What is it? No idea. Whenever you touch something hot, you detect that sensation. That sensation goes to your spinal cord because those are simple things, no? Right? Goes to the spinal cord only. Brain එක උඩට මෙන්නේ නැහැ. In the spinal cord, there are interneurons. So the interneurons in the spinal cord can take a decision, and that decision will be sent to your bicep. So you will take the finger away from the whatever hot object. This is called reflex arc. Ayyo. Right? Kati doide. Right? It's called reflex arc. That is the basic structural, sorry, the basic functional unit. Structural unit is the neuron, yes. Structural unit is the neuron, that is correct, but not the functional unit. Okay. The most abundant cell is the neuron, no, right? Neurons are very big, yes, but not the most abundant cell. What is the most abundant cell in the nervous tissue? It is the glial cells, neuroglia, right? You have so many glial cells. Matakai, the nervous tissue, right? 4 weeks ago, right? Tissue seminar, okay. Neuroglia participate to provide strength? No. They regulate, insulate, nourish, and what else? Nourish, insulate, regulate. There's another function. Modulate or whatever, something, right? Those are the functions of neuroglial cells, but not strength or something, right? Neurons are bundled together into a nerve. What do you think? Neurons are bundled together into a nerve. The axons of neurons are actually bundled into a nerve. Axons. What do you mean by an axon? What do you mean by the axon of a neuron? So I'll show you. What do you mean by the axon of a neuron? So if you take a typical neuron, like this, no, okay. So you can see this main structure is the neuron. This is the neuron. These are all different types of glial cells. Maybe green, blue, purple. These are all different types of glial cells, right? So you can see in this, so this is probably a motor neuron. These are all inputs into the neuron. Those are bringing signals towards the nerve cell body, which is this one. Okay, they are bringing signals towards the nerve cell body. So they are called dendrons or dendrites. Smaller ones are dendrites. This is the nerve cell body. You can see arising from the nerve cell body, you have a big output and a very long arm. You can see, sorry, this one. If I zoom out and turn it this way, hold on. Can you see this structure? From here to here, you have a very long process that is arising out of the nerve cell body that is called the axon. That is the axon of the neuron. So axon takes signals away from the nerve cell body. So these axons bundle together to form a nerve. A nerve is like you have sort of wire cables, no? Right? Bringing internet into your house, you have fiber optic cables, electricity cables, all those are there. No? So these nerves are sort of that sort of wire bundles that are formed of these many axons. What is the only place in your body you can touch a nerve? You can physically palpate a nerve. Otherwise, nerves are running sort of deep in the sort of the body. You can touch a nerve here. This is called the ulnar nerve. Ulnar nerve එක මෙතන අහුව. Got it? Current එකක් තියෙනවා. මේ sort of this part of the palm. Have you felt, right? So that is one of the places you can feel a nerve. So axons bundle together to form a nerve. Okay. where's my edits? Hold on. What happened? Oh, yeah, sorry. Right. So, neurons are bundled together into a nerve. Neurons are bundled together. That is, I mean, neurons are itself not bundled. The axons of the neurons are bundled together into a nerve. Neuroglial cells can divide. That is actually correct. Okay, neuroglial cells can divide. That is the correct statement. Uh, okay. So we can take it as the correct answer because this is not. You can correct this. Neurons are bundled together into a nerve way. Axons of neurons are bundled together to form a nerve. Okay. Okay. So your neuroglial cells can divide because they are the sort of the supportive. Cricket team there are some there's a lot of people going behind. There's a doctor, there's a physiotherapist, there's a nutritionist, there's a coach and everything. Right? When the sports team is going, even a single sportsman. Okay. So the neuroglial cells are there for the support. So the supportive cells can divide. But you know this, right? Your nervous cells, they are unable to divide. They do not divide. They are what we call as permanent tissues. They cannot divide. Okay, throughout life, the same number of nerves remain as you, I mean, as what you have at birth. Okay, they do not divide and grow again and again. Okay, which of the following statements regarding Holozoic? So, we are coming to a bit of animal form again. Holozoic mode of nutrition. What is the definition of Holozoic nutrition? They ingest food into Holozoic nutrition means they ingest food can into the alimentary canal. They have a sort of a food processing tube that is designed to digest food and extract the nutrients. So the food, if you are a holozoic animal, that means you eat food into your sort of alimentary canal. There are different modes of nutrition in different animal species according to the food and environment. Yes. They are asking incorrect one. The last two process of food processing occur after food digestion. What are the steps in food holozoic nutrition? What are the steps in food sort of holozoic nutrition? You have can. What are the steps? Ingestion. There is the eating, digestion, breaking down into smaller molecules, smaller pieces, so that they can be absorbed. Then once you have absorbed, they are used in different body functions, right? Repair, energy production, whatever. Okay. I'm sorry, digestion and absorption missing, right? Digestion, you have absorption. Okay, digestion, absorption. Then after absorption, you are using them for different functions, assimilation. Then finally, you are removing whatever you don't need, that is elimination. To be honest, this assimilation is not part of new digestion, right? It's not part of nutrition assimilation, but because it happens everywhere else in the body, I mean, it happens throughout the body. The last two processes of food processing occur after digestion. What do you think? Yeah, I mean, after food digestion, you can say food digestion. So there's actually three more steps, but let's keep it. There's a more incorrect one. Absorption of digested nutrient products occurs actively and passively. So yeah, absorption occurs actively and passively. No, so if you take things like your fructose, they are passively absorbed. If you take things like glucose, amino acids, vitamins, other things, they are absorbed actively, I think we discussed just last week this absorption. All steps in holozoic mode of nutrition in animals occur only in alimentary canal. What do you think? Yes, holozoic nutrition means you take it into a alimentary canal. No? There is the definition. Holozoic nutrition means you take it into alimentary canal, right? You have to take it into a certain system in your body that is designed to do those things. Okay, so always you have an alimentary canal involved. So that is correct. Defecation is not considered as excretion, that is correct. It is not a method of excretion. Excretion we consider as to be the removal of products of metabolism, right? You produce waste products as byproducts of metabolism, mainly nitrogenous waste products. Removal of those is excretion. What is defecation? That means you are removing your fecal matter. That is what? That is you are just getting rid of the undigested food. That is not excretion. So that is also correct. So we make the last two processes, there are three more processes to occur after digestion. So answer number 2. Okay. Right. Question number 12. Select the correct statement regarding ascent of xylem sap. Xylem ascent. Right. We are jumping again to a bit of plant form. Water and minerals are transported in xylem by diffusion? No. Diffusion is a short distance transport. It's a short distance transport. So xylem is a long distance. I mean, it has to be long distance because why? A tree can be sometimes even several hundred. I mean, about several sometimes 100 meters or even taller. So it has to be long distance. It has to be done by bulk flow. So diffusion is short distance. So that is not enough. Okay. Diffusion is short distance and that is not enough. Okay. Water molecules are attracted only to water molecules in this process. This is basic our sort of functions of water. The functions of water. Right. What are the forces or the, I mean, cohesive and adhesive forces both are involved in xylem ascent. No? So what do you mean by cohesion? That means water, water. Hurry, the make and ek. Here we are talking about cohesion. But do you require adhesion? Yes. I mamme explain karanne. So for example, imagine the xylem vessel is like a tall tube or a well. So you are inside that well, someone is giving you a hand. That is the water molecule that is on top of you. Alright. So that water molecule by cohesion he is trying to pull you up. But you can help him to stop yourself from falling down further by putting your legs right onto the two sides of the well. You can uh, put your legs on the two sides and stop yourself from falling down further. Okay, that is done by adhesion. Okay, so you need both cohesion and adhesion for xylem ascent. Okay, right. Xylem sap is transported under tension. Yes, it is under negative pressure, right? It is under negative pressure, which is what we call as a tension, right? Because it's an upward pull, no? So pull legak is what we call as tension. Okay. Xylem sap. Okay. Xylem sap is transferred even through symplast to the shoot system. Xylem, does it ever travel in symplast? Xylem, does it ever travel in symplast? What do you mean by symplast? What do you mean by symplast? Symplast means it involves what? Symplast involves crossing of a plasma membrane at least once. Crossing of a plasma membrane at least once. So does that happen in so we have found the correct statement, no? Right? We have found the correct statement, right? So answer number 3. Even through Symplast, no, because Symplast involves crossing at least once through a plasma membrane. Okay, crossing at least once through a plasma membrane. So if you have a plasma membrane, that means it's a living cell. Xylem vessels or xylem tissue is a dead tissue. Therefore, it doesn't involve in any living tissues because xylem vessel elements, tracheids and all are dead tissues. Okay. It doesn't depend on the amount of water vapor in the external environment. What do you think? Yes, it depends because why? Who provides this transpiration pull? Who provides this transpiration pull? Right? Who is the upward pulling force? Who is the upward pulling force? That is transpiration. Transpiration. When water is lost from the leaves, that water will be replaced by some more water that is coming up. Therefore, transpiration has a direct effect on what? Xylem acid. Transpiration depends on what? Amount of water vapor. What do we call this? The amount of water vapor. Physics world have you learned this? Humidity. The relative concentration of water vapor in the environment. We call it humidity. Okay. So answer, I think answer 3 is the best. Okay. Clear? Okay. Right. Question number 13. Um, okay. These are about plant nutrients act as a cofactor in chlorophyll synthesis. You can take boron. I think I can remember boron is a cofactor. Activator of many enzymes, isn't it magnesium or zinc? Chlorophyll synthesis, you need iron. Isn't it answer number 1? Is there any other answer that is matching for this? I can't be bothered at this point to study plant minerals because that is your job. Okay. Now I am lucky enough not to study. So iron you can take for chlorophyll synthesis. Yes. My doubt is activator of enzymes. Yeah, you can take magnesium also. Boron, magnesium, and iron. So answer number 1 and 4 both correct. Okay. Magnesium is an enzyme activator as well. Okay. Magnesium is an enzyme activator as well. Okay. Understood? Clear? So yeah, straightforward question, very easy. Correct statement nahanne gametophyte of Nephrolepis is a photosynthetic dioecious plant. What again? Correct statement. Matakal then Nephrolepis life cycle. What do you think? In Nephrolepis, you have a heart-shaped photosynthetic hari, sort of a it's a macroscopic structure. You can see there are gametangia. Okay. So is it a plant? No. It doesn't have any proper tissues. We call it, it's simply a thallus. It's a ball of cells. It's not a proper plant or a proper, it's not a proper plant because it doesn't have proper tissues. It is just a ball of cells that we call as a thallus. So that is incorrect. No. So male gametophyte matures within the microsporangium of Selaginella. What do you think? No, you release the immature male gametophyte, neither, right? Microsporangium produces microspores. You release the immature male gametophyte in the microspores. So that is also false. After the development of the female gametophyte of cycas, the remaining megasporangial tissue is nucellus. Yes. Okay. Nucellus provides nourishment. Okay, new cellus provide nourishment to the female gametophyte. Okay, new cellus provides nourishment to the female gametophyte. These are plant life cycles. Okay, in cucurbita, right? Okay, pollen tube is formed by the generator sieve. This is a bit out of syllabus, but we can come at. We can arrive at the correct answer, that is answer number 3. So don't think about it, it's a bit not in the syllabus. Foot of the Phogonatum sporophyte absorbs water from the soil. No, neither. Right? If you can remember Phogonatum life cycle, can you remember? Sporophyte is non-photosynthetic, of course. Right? It is attached to the female gametophyte. It is attached to the female gametophyte by its foot. Neither. Its foot is involved in absorbing nutrients from the female gametophyte, right? What are the three parts of the male spore? What are the three main parts of Pogonatum sporophyte? You have a foot, seta, and a capsule, right? Foot is connected to the female gametophyte. Seta is sort of like a stalk. Capsule is the sporangium. So the sporophyte of Pogonatum is very simple, you know. So foot is not on the soil; it is connected to the female gametophyte. Okay. I always recommend that you study these while by remembering the life cycle, by remembering the image. It is going to help you a lot when answering MCQs. So foot absorbs water from the soil. No, it is connected to the female gametophyte. Right, question number 15, plant hormones that delay leaf senescence and promote leaf senescence respectively. Hari. So delaying leaf senescence, making a vibe again, right? Delaying leaf senescence is what? It's something good, no? It is something positive. Leaf senescence means the leaf is getting old. So it is something positive. So if you are delaying senescence, that is something positive. Promoting leaf senescence, that is something negative, no? Right? Something, you are doing something negative. So, what are the positive vibes in plant growth substances? Abscisic acid, gibberellin, cytokinins, gibberellins, and yeah, those are the two answers that are saying delaying leaf senescence possible. No, promote leaf senescence could be abscisic acid, not this, neither. Oxine is a positive one. So answer number 5 is out. So who promotes, delays leaf senescence? So because we have the same answer at the back now, right, as the answer number 2. Who delays leaf senescence? It is cytokinins. Because why? Gibberellin usually involves in sort of your sexual things, no? Right? Maturation of gametangia, seed germination, aromation, right? So reproductive functions are mainly regulated by gibberellins. Okay. So yeah, dioecious means unisexual or bisexual. Dioecious means there are two plants. Dioecious, that means, what do you mean by two plants? So there's one male plant, there's another female plant. So that means sexes are divided unisexual. Right? Okay, so we'll discuss question number 16. We have some more MCQs. right? We'll stop with question number 16, and we'll go ahead with the discussion of the remaining MCQs and the structured essay next week, okay? Because I'm getting messages. There's in certain places there's a power cut, okay, right? So let's see. I mean, we'll continue the discussion next week. Right, let's discuss question number 16 and stop for the day. Okay. Correct statement regarding regulation of digestion. Oh no, this one is going to happen on this Sunday. Right. So we are going to talk about regulation of digestion this Sunday. So I'll leave this question. Shall we discuss one more like this one? Question number 17. Regulation of digestion. After this week's theory class, you can do it, but I anyway included it. Okay. Uh, right. Question number 17, there's a small error, neither, right? The question is not there. Okay, so you can cut it. Yeah, there's a small error with question number 17. Okay, you can cut it because the question has not been included. Okay. Features of cells that are labeled as A, B, C, and D, right? Large central vacuole is present. B, presence of a comparatively thick primary cell wall. C, central nucleus is present. Contain pits. Hurry. Okay. What are the options? Large central vacuole, probably in parenchyma, never in meristems. Neither meristems do you have a central vacuole. No, meristems are very metabolically active cells. So they cannot have a large vacuole, they are dense cytoplasm. So you can have no meristems. Meristems they never come out. Phloem parenchyma could be, xylem parenchyma could be. Comparatively thick primary cell wall, yes, collenchyma neda, right? Collenchyma තමයි usually. Epidermal cells not very thick. Central nucleus is present, okay? Central nucleus, yes, usually in meristematic cells, neda? Meristematic cells, so there is vascular cambium. Presence of pits in secondary walls, tracheids. Answer number 5, neda? Okay, presence of tracheids. I mean, in tracheids you have pits because through the pits water travels. Okay, any questions? We'll continue the discussion then next week. Okay, so in the 2020 in your theory this Sunday we are going to finish nutrition probably. Okay, then we are going to discuss. I mean, there are some structured essay questions we need to complete from nutrition as well. Then there's an upcoming exam, so keep that in mind. I won't include animal form in that exam. Up to the end of plant form, everything will be questioned. Okay, right? So I'll mark your papers and give them to you. Question number 17. What happened there? I'm thinking. Characteristics of living organisms. Yeah. So. Okay. Yeah, I'll modify and send to the group. Okay. I'll modify question number 17 and send to the group. Okay. Right. Okay. Right. Okay. Okay. So we'll stop for the day. Any questions? Right. So join next week with the discussion of the second part. And okay, be safe from the adverse weather and keep studying. Okay. Then good night. Yeah. Thank you for joining. Okay. Okay. Thank you. Right. Okay.